So a lot of this stuff in this presentation you already know. I'm going to go through it fairly quickly, and then you can check in with me if you've got questions. So what are your cells made of? They're made mostly of water. It's the most common molecule in all of your cells. Water is inorganic. It makes up 60 to 70% of your mass, but it's also 99% of your molecules. So 95 to 99% of your molecules, if you count them, are water. The rest of them are organic molecules. So that tells you that these organic molecules, because they make up 30% of the mass of your body, but only 1% of the molecules, that tells you that they are way bigger and way more massive than the water molecules. The definition of an organic molecule is that it always contains carbon and hydrogen. It may also contain nitrogen, oxygen, phosphorus, or sulfur. So I use the mnemonic SPONGE to remember all the possible elements in organic molecules. You should study and be familiar with these different functional groups. These are just collections of atoms that you find frequently in organic molecules. Anytime you see an R, that means it's variable. So this OH is the hydroxyl group, and it's attached to something, right? Some other larger molecule. And so you should be familiar with these. Hydroxyl, methyl, carbonyl is the double bond to an O, carboxyl group, O, OH. Also be familiar that this is acidic because often in solution it actually loses this hydrogen, so it becomes O negative. And so it donates a hydrogen solution, so it's an acid. Amino acid, or the amino group, is actually basic because it can take on an extra hydrogen, so it takes a hydrogen away. Phosphate group is basically a P surrounded by O's and sulfhydryls S attached to an H. So be familiar with those. There are 4 major categories of organic molecule: carb, protein, lipid, and nucleic acids. We're really going to focus on these first 3, and nucleic acids come in their own unit later on. Carbohydrates, you should know their general structure, right? They contain C's, H's, and O's, and there's always a 2-to-1 ratio of hydrogen to oxygen. I always kind of say that they are carbons that have been hydrated. So there's 2 H's and 1 O is H2O. So it's a bunch of carbons with water attached, right? These are hydrophilic. Different categories: mono, di, and polysaccharides. Mono means 1, di means 2, and poly means many. So you should be familiar with these 3 categories. You do not, however, need to know that sucrose is made of fructose and glucose, for example, but you should know that it is a disaccharide, as well as maltose. And you should know starch, glycogen, and cellulose are all polysaccharides. The monosaccharides, the monomer, and the polysaccharides are the polymers. So dehydration synthesis or degradation hydrolysis. So dehydration synthesis is when you take monomers and you join them together to make a larger molecule, right? Every time you do that, you have to remove a water, you dehydrate it, right? So in this case, we removed a water for each new bond we're making, right? And degradation hydrolysis is when you are doing the opposite. You're adding water in to split apart a larger molecule, right? You can take a look at these examples. I'm not going to spend time on this right now. And on the next slide, I'll show you the answers. Okay, this is just a diagram picture showing you where those OH and H are removed from. So it's removed here, and then there's always an oxygen between the 2 monomers when you're looking at carbohydrates. Okay, so you remove the water to allow these 2 to join together. Then we have our category of lipids. Lipids are also made of C, H, and O, but they are not in a 2-to-1 ratio. There's actually way fewer oxygens in lipids than there are in carbs. They are hydrophobic because a CH bond is hydrophobic. It's a non-polar bond, and lipids are mostly made of C's and H's. So they do not mix well with water. They're non-polar, and they're not true polymers. They're not one repeating unit over and over again. Examples of lipids, so fatty acid molecules are lipids. You've got a carboxyl group and a hydrocarbon chain. The hydrocarbon chain can be saturated or unsaturated. If it's unsaturated, it has a double bond, 1 or more double bonds. If it's saturated, it's got all single bonds. Fatty acid molecules, as you can see, the saturated ones are generally straight and the unsaturated are kinked. but it really depends on the position of those hydrogen atoms adjacent to that double bond. If they're on the same side, then that's called a cis orientation of this fat, and there is a bend or a kink in the chain, and that makes them liquid because they pack less densely. If you position the hydrogen on the opposite sides of that double bond, this becomes a trans fat, and the trans fat is actually straight and appears more like the saturated fat. So trans fats have properties that are more like saturated fats. They do not exist in nature, really. They're man-made. They're called hydrogenated fats, and that's what they look like. These are solid, and they pack very densely together. These are not good for you. A triglyceride is what we think of when we think of fat in a food, right? They are mainly for energy storage. They are made of 1 glycerol and 3 fatty acid chains. So here's the glycerol, and here are the 3 fatty acid chains. If there's even one double bond, we call that unsaturated. You can call it polyunsaturated, monounsaturated, depending on how many unsaturated fatty acid chains there are. Another example of a lipid is a steroid. They have these rings of carbon. It's different than a carbohydrate ring of carbon, though, because they're all interconnected. They're all sharing carbons at intersections. Notice that in a line diagram like this, every intersection is a carbon, and then you fill in hydrogens, right? So this whole structure is just C's and H's, and there's only 2 O's. Okay, cholesterol is an example of a steroid, and sex hormones like testosterone and progesterone are examples of steroids. Phospholipid is another kind of lipid. They basically look like triglycerides, except for instead of a 3rd fatty acid chain, which would be here, there's a phosphate group attached to it. And depending on what else is attached to the phosphate group up there, they've got different names. This is special regions, some like sphingolipids, and there's different names for these different phospholipids. But generally, phospholipids are a major component of cell membranes, so they're really important. And they usually have these kinked double bond tails, so that it doesn't get too solid. Because if it gets solid, then the membrane will be solid, and things will not be able to move into and out of the cell. This is kind of a cartoon drawing of how we draw a phospholipid. We'll look at that in our next unit. But they orient themselves with the heads on the outside of the cell and the inside of the cell, and then the hydrophobic tails are kind of in between those heads, forming a barrier. Atherosclerosis is the most common cause of death in the U.S. This is happy arteries are elastic and unclogged. So what increases risk for atherosclerosis are high blood pressure, lots of LDL cholesterol that generally what's doing is putting the fats in your blood vessels, it's storing them in the wrong place. Cigarette smoking increases risk of this, low exercise, heredity factors, and eating too much of the bad cholesterol, too much bad fat. That basically what's happening is you have these blockages in your blood vessels, so the blood can't get through. Now, if there's a blockage in the blood vessel to your heart, that may end up in a heart attack, right? Or if there's a blockage in blood flow to your brain, you may end up with a stroke.